A very warm welcome to each and everyone who is listening to this video or who is watching this video. So I'm going to discuss on a topic of parasite. So one of the name of the parasite, Giardia Lamblia. So I'm going to talk about this Giardia Lamblia. So this parasite, it is one of the earliest protozoan parasite to have been recorded. It is a flagellate. So this flagellate was first observed by a Dutch scientist whose name is Antony van Leeuwenhoek in 1681 in his own stool sample. And how do we got the name of this parasite, Giardia Lamblia? It is named Giardia after Professor Giard of Paris and Lamblia after Professor Lambel of Prague. And this parasite, it is a common protozoan pathogen and it is worldwide in distribution. And the habitat of this parasite, it is mostly found or it lives in the duodenum and upper jejunum of the human body. Next we have is the morphology of Giardia Lamblia. This Giardia Lamblia, it exists in two forms of morphology, that is the trophozoites and the cysts. So you can see the picture of the trophozoite and cyst, it is provided here. So this trophozoite, it looks like a tennis racket. If you see the shape of this trophozoite, it is similar to a tennis racket or heart shape or pyriform shape, and it is rounded anteriorly and pointed. Trophozoite, it measures around 15 micrometer and 9 micrometer wide and 4 micrometer thick. Dorsally it is convex and ventrally it has a concave sucking disc which helps its attachment to the intestinal mucosa. Then this trophozoite is bilaterally symmetrical and they possesses one pair of nuclei, 4 pairs of flagella. They have the pharoplast which from which the flagella arise, and they have one pair of exostyles running along the midline. They consist two sausage-shaped parabasal or median bodies lying transversely posterior to the sucking disc. So this trophozoite is motile with a slow oscillation about its long axis, often resembling a falling leaf. So that's all about the trophozoite. Now the next morphology we have is the cyst. It is the infective form of the parasite. This cyst is small and oval. The size, it measures about 12 micrometer into 8 micrometer, and it is surrounded by a hyaline cyst wall. Its intestinal structure includes two pairs of nuclei grouped at one end. A young cyst may contain only one pair of nuclei. Then this cyst also has a exostyle that lies diagonally, forming a dividing line within the cyst wall. And the remnants of the flagella and the sucking disc may be seen especially in the young cyst. Next we have is the life cycle of Giardia Lamblia. So this life cycle, it passes the cycle only in one host, and the infective form, like I have mentioned, it is the cyst, not the trophozoite. And the mode of transmission of this disease, that is giardiasis, it is or the mode of transmission of this parasite, it is through infected or ingestion of contaminated food and water, or it can also be through direct person to person contact or person to person transmission, which may occurs in children or male homosexuals and mentally ill persons. So this the cycle will be like I have said, it will start through by ingestion of contaminated water and food. So the cysts will be present in this contaminated food and water. So the cyst will travel through the esophagus, it will reach the stomach. After reaching there, it will bind to the mucous layer of the small intestine or the duodenum. Then in the small intestine, one cyst released to trophozoite, and this process is known as excistation. And this cysts multiply through longitudinal binary fission inside the small intestine. Then now the trophozoite attached to the surface of small intestine, causing abdominal pain, cramped, or dysentery. Now the trophozoite will move to the colon and produce cysts, or they will produce new cysts. The process is known as encystation, and these cysts will pass now in the feces or the stool. Then these cysts are very resistant, and they are the infective form, and they can survive for many months in, especially in the warm climates or warm area. So that's the life cycle of Giardia Lamblia. Then the pathogenesis we have for Giardia Lamblia. Giardia Lamblia, it will cause a disease known as giardiasis. This Giardia Lamblia is typically seen within the crypts of duodenal and jejunal mucosa. It does not invade the tissue but remains tightly adhered to intestinal epithelium by means of the sucking disc. They may cause abnormalities of villus architecture by cell apoptosis and increase lymphatic infiltration of lamina propria. Then variant specific surface proteins or VSSP of Giardia play an important role in virulence and infectivity of the parasite. And often this parasite, they are asymptomatic, but in some cases Giardia may lead to mucus diarrhea, fat malabsorption, which is also known as steatorrhea, dull epigastric pain, and flatulence. And the stool will contain excess mucus and fat but no blood. Then this parasite in children, it may develop chronic diarrhea, malabsorption of fat, vitamin A protein, sugars like xylose, disaccharides, weight loss, and Sprue-like syndrome. Sometimes or occasionally, this Giardia may colonize the gallbladder, causing biliary colic and jaundice. Then the incubation period for this parasite, it is variable, but it is usually about 2 weeks. So that's all about the pathogenesis. Then coming to the lab diagnosis of Giardia Lamblia. So the specimen we mostly preferred here is the stool sample, since it is related to the infection or the disease caused in the small intestine. So the sample will, or the specimen mostly preferred is the stool sample. So there are lots of methods we can perform to detect this parasite. When it comes to the lab diagnosis, you can perform stool examination, entero test, or which is also known as the string test. We can also perform the serological test, that includes antigen detection and antibody detection. Then we can also perform molecular diagnosis by DNA probe or PCR or polymerase chain reaction. So mostly out here I'll be discussing about the microscopic part or the microscopy examination and the entero test or the string test. So the stool examination, this giardiasis can be diagnosed by identification of cysts of Giardia Lamblia in the form stools and the trophozoites and cysts of the parasite in diarrheal stools. On macroscopic examination or the physical appearance of the stool sample or specimen containing this parasite, that is Giardia Lamblia, may have an offensive odor pale in color and fat will be present, and they will float in water. That is the macroscopy examination of the stool sample. Then on microscopic examination, we will see the cysts and trophozoites in the diarrheal stool by saline and iodine wet mount preparation. Often multiple specimens need to be examined, and concentration techniques like formal ether or zinc acetate can also be used. And in case of asymptomatic carriers, we can see only the cyst, not the trophozoite. So that is all about the microscopic examination of Giardia Lamblia. Coming up next we have is the entero test or string test, or it is also known as gelatin capsule test. So this test, it is a useful method for obtaining duodenal specimen. Okay, out here a gelatin capsule containing a nylon string with a weight attached to it, and the free end of the string is taped to the person's cheek, and the capsule is swallowed by the patient. Then the capsule will dissolve in the stomach, and the string, due to its attached weight, moves down to the duodenum and jejunum. Now the string is allowed to remain in the intestine for at least 2 to 4 hours, or sometimes overnight, while the patient is fasting. Then after that, this string we will remove the string, and the bile stained mucus is then collected on the glass slide, and it is examined on microscope for the presence of trophozoites, or for the detection of trophozoites. So that is all about the Giardia Lamblia. Thank you.